Welcome to the IDEA National Resource for Quantitative Proteomics. I'm Stephanie Byro, and today I want to talk about ProteoDA, which is an R package our team developed to help analyze mass spectrometry datasets. This R package was really designed for biologists to be able to reinvestigate the results coming from a mass spectrometry experiment. And so the R package is published in the Journal of Open Source Software, as you can see here, but all of the code for the package is freely available in GitHub under Biome Lab ProteoDA. Part of this package, I will show you the GitHub will show you how to install the package, but I will go through a detailed example of how to actually run a project through this R package. But before I do that, I want to go over the main workflow that's associated with this package. So there's 3 file requirements needed for data import. One is your protein intensities. 2 is the protein annotation information, including UniProt IDs, gene names, protein descriptions, any sort of protein annotation that you may have with your data set. The metadata is the list of samples that are part of the project and their grouping information. So whether they're control or treated, any grouping information. Also, you want to include any batching information, whether samples are collected at different days, they belong to certain batches, also gender effects, any other factors that you're going to analyze in your data set should be included in this metadata file. So once you have your 3 input files, those will then go into a DA list of the R package. This is a function of the R package that will allow you to utilize all of the other pre-built functions throughout the process and makes the analysis smoother. So the DA list is based on differential abundance because we're measuring protein abundances. Once you have the data incorporated into a DA list, the first step is pre-processing. So we want to look at filter. We included multiple filter functions in case you need to remove any outlier samples, if you need to remove any certain proteins, proteins by annotation, proportion. There's multiple different filtering options to make your analysis smoother. Once you have filtered out the proteins or the samples, you can then write a normalization report. Part of this normalization report includes the package ProteoNorm, which analyzes 8 different normalization methods to allow you to investigate the variability and the correlation of your sample replicates within each group. And this will allow you to evaluate the better method for your particular data set. Once you have the normalization report, you will then choose one of those 8 methods that are included and use the normalized data function in order to produce that final result. So once the data has been quality filtered, it's normalized, you can then write a final quality control assessment report, including Violin plots, PCA, cluster dendrograms, check out your missing values, how many missing values might be included, to see if you need to go back to the filtering steps and redo some processing of the data. Once everything is processed, normalized, then we perform statistical analysis. The statistical design is based on the Lima R package. So we've included an add design function, which allows you to spell out the formula that you would like to use for your linear model, such as either the no-intercept model design or the intercept model design, and then you can list and tell it what column in your metadata is the grouping column. You can also, this is where if you have a batch effect that you're trying to remove, you can say group plus batch, and that will be included in the model matrix. Add contrast is an option where you can tell it specifically how you want to define your sample comparisons. How do you want to how do you want to analyze the fold change associated with your contrast with your project. And there's two ways of adding contrast, either directly into R code, or you can just submit another CSV file that lists the different comparisons that you would like to analyze. So once the design is set up properly, then we run a Lima fit model, and we have functions to extract all of your data results into either tables or plots. And so you will have both tables with all of the information and all the statistical values for each protein, but then those tables are also provided in interactive reports using HTML web browser. So then you can actually click on each of the proteins that are listed in a volcano plot, for example, and you can see the information about this is the name of the protein, this is how abundant it is, and you can also will see a separate plot show up that shows the each individual sample replicate and their abundance values. So now that I've given a brief overview of what this package is, I will now show you the example that is included in the GitHub of how to run this using RStudio. So we will go out of, and the GitHub, if you are not as familiar with R, we did include how to install this package directly. So I open up RStudio, and this is an example ProteoDA script, R script. The very first command here is to install packages, the dev tools. Because we are installing this from a GitHub repository and not directly out of CRAN or Bioconductor, you'll need to have the dev tools installed. And currently for this example, I am running R 4.3.2 version. And yeah. So I do have in my example a couple of extra packages that needed to be installed. This was related to a Windows computer that was missing some basic packages with R. So these are not typical ones that you would have to install, but if you do run into some troubleshooting, this is what helped fix the installation on a Windows machine. I'm currently running this on the Mac for this example. So we want to run DevTools install GitHub BioRamLab/ProteoDA. If you run it with the dependencies equal true, it should install the packages required for this package to operate. And then build vignette set to true will also generate an HTML file that will you can open in your web browser, and it will have all this code that I'm about to show you in this vignette. However, sometimes just a note of caution, if you run into some install issues, if you set build vignette to false, it will probably load in better. I have had a little bit of trouble on various versions with the building the vignette. Okay, so the first thing we want to do after the package is installed is, of course, load our library. So library ProteoDA. And then if you were able to build the vignette, you can browse the vignette, and this will give you a new browser and open up to an HTML file. And again, this is where you see how to install the package, the code, and now it's going to give you more detailed explanation on how do we import those 3 files that I talked about that are required for this this package to run. So it will give you more details, explanations, how to run it, and then it will also include the code as showing how to run this package. So the vignette is quite useful. So going back to RStudio, the first thing that I have is an example data set from a melanoma study that we're using to show the functions in the package. So you would load this CSV file from the system file included in the R package as the DIA data and then the meta file. So I'm loading this in as my input data. If you have your own file, the CSV file should include the uniprot_ID. This column header is required for your input data. This is very important because it's going to set the row names in the previous and the next steps of the package. So you do have to have uniprot_ID all lowercase specifically in one of the columns in your data set. In this case, I have the uniprot IDs, protein information, which is the FASTA header, some accession number, molecular weight. There's a few other additional columns within my data frame that are related to the protein annotation, which I mentioned previously. And this will make more sense here momentarily. But we have the protein annotation information followed by each of the samples. So our samples start with pool_1, pool2, pool3, followed by the samples. In our experiment, we ran a pool sample that is a mixture of all of the samples included in the data as a way of testing batching. So we have pool samples, this samples 1, 2, 3. This particular data frame, any missing values that are included in my data set are listed with zeros in the data frame. This is also very important to pay attention to because some software will put an NaN as the missing value instead of 0 or Na. ProteoDA currently just accepts zeros are an Na value, not the NaN. So you may have to replace those if that's an issue in your data set. The other thing to point out, so this is our input data. Let's load in our sample metadata file. And if we do this, we'll look at our sample meta information. Now you'll see I have an extra column in here labeled data column name. And the reason I do this is so I know which column in my sample meta sheet is going to match the column names in this data input. So it's very important that the column names here of your data matrix matches the row names of the sample metadata. So this is going to become how we are matching the sample intensities back to its sample information. So that is a very important grouping in your data. So we have the input data, we have the sample metadata. And remember I mentioned that you actually need three tables to go into the DA list object. And so what we're going to do next is actually I'm going to split this input data frame out to where I only include the sample intensity data and I remove the protein annotation information into a separate table. So to do this, I've got I'm going to call it the intensity data, which is my input data. Take all of the rows and take columns 5 to the end of the data frame. So we'll make our intensity table. This now includes only the samples that are in the data. The annotation data, this one will now include only the annotation information. And so we want to check our row names of the sample metadata. And as you can see, when we loaded it into R, it doesn't have the row names labeled. It's just one to the end. And so we have to make sure that the row names of our sample metadata table match those column names of our data table. So we're going to rename the sample metadata based on our sample metadata sheet data column name column. And then these names will match our intensity data. So when I do that and I rerun row names, now instead of 1 through 17, you actually see the names of the samples that are listed in your metadata sheet. So if we click on metadata, instead of 1, you now have your row names associated. Once this is really the hardest part of running the package, is formatting your data to fit within the structure of this R package. Once that is done, you can create this DA list. We call it raw because we haven't done any processing. It's just the raw data. And we click run. If you pass this without any errors, then you should be good to go for the rest of the functions. So as I said, in our example, we have a group of samples called pool, which is just the pool of all the samples kind of for a technical look at mass spectrometry data. Well, we don't want to analyze that in our differential abundance. So what I'm going to do is actually remove anything that has a group. So in my metadata, I have a column called group, and anything that's listed as a pooled sample will get filtered out so that I'm not including those samples in my normalization. So we'll filter the pool samples. It'll show you how many samples at the bottom are removed and how many are kept. And then another important thing is converting those missing values. So when I imported this data, my missing values included a 0. And in mass spectrometry data, 0 just means it was not identified. Doesn't mean it wasn't there. Just a little caveat. So for doing the lemma analysis, when I do my fold change, I actually don't want to include those zeros in my analysis. So lemma has a function to omit NAs. And in order for us to use that omit NA function, we'll need to convert our zeros to an NA. So this is what I'm doing here is I'm converting those missing values from a 0 to a missing value for the lemma analysis later on. So now if we make a new object called filtered samples, this filtered samples will have less samples in it than we started. We will have our annotation information, all of our data, metadata, everything is associated in this same object. So the next step, this is again a decision that you can make in your analysis. So just a general rule of thumb, I usually filter and I usually require 2/3 of the number of replicates that you have in one group to have a value, at least in one of those groups. Meaning I don't want to have any proteins in my data frame that are just have 0 values across every sample in your data set. So how much of those proteins that are missing a value you want to filter, you can control with these parameters. So the minimum replicates means I'm requiring at least 4 replicates out of this group of 8. I think there's 8 samples in each group in this example. Don't quote me on that. But so say 2/3 of your replicates out of one group. If you wanted to require that both groups had to have, then you would just make this 2. So this gives you flexibility on how much lower abundant proteins you might want to include in your analysis, and you can kind of go through this multiple times. So if I use this setting, it will tell you how many proteins are going to be removed from your data set. So in this case, 203 proteins were removed, and that left us with 8,886 proteins still left to be analyzed. So that's a good number. So we've filtered out proteins, kind of dealt with our missing values, and now we're going to run our protein normalization report. So here we have the grouping column. Again, make sure whatever is listed in your sample metadata, this is the value that you would want to put as your grouping column. You can tell it where you want to save your output. You can label the file name if you need to change it. Be careful with the overwrite option. Right now I have it set to true because I've run this multiple times. And so let's go here, and before I run this, I do want to check what directory I'm sitting in. Yes, okay, so I know where this is going to save. Okay, so this is going to generate a normalization report using ProteoNorm that was also published by myself and my team. So we just incorporated ProteoNorm into ProteoDA to make it much simpler. And then once this completes and generates our report, which it'll show up as a new folder and a new PDF file, we can open this here. computer's taking a moment. And so what you will get is a description of what the top plots are showing you are variability plots: pooled coefficient variation, mean absolute deviation, pooled estimate of variance. So what we're looking at is the variability among the replicates within each group. So each dot that is on the plot represents a group. In this case, we have a cancer versus normal. And so there's 2 dots and showing you your variability. The other thing of interest is correlation. So correlation, we want our replicates within a group to be highly correlated across the board. So in general, if you have really clean data, it's cell lines in particular, you may not see much difference between each of these methods. The log2 is the unnormalized. It's only log2 transformed. So you can see how normalization actually reduces the variability within the data set on these top plots. But this will help you to select your method of choice to do the normalization. The log2 ratio is the what happens when we do the fold change calculations. We're making this assumption in protein data that the majority of the proteins don't change. And with that, you don't want to have your normalization method to start shifting your log2 ratios negative or positively. That density should be highly centered around 0. This particular data set has a little more variability in it than a lot that we see, but it's due because it came from frozen paraffin embedded tissues. So you would expect to have a little bit more variability in tissue samples compared to, let's say, cell lines. But overall, in this normalization, when the values look very similar, I tend to go with cyclic low S. It seems to be a really nice performing method, and it's also part of the Lima package. When you have really low abundant molecules with more missing data, the variance stabilizing normalization, or VSN, tends to perform better. That's just a general. So you have your normalization report. We then can go and run our normalized data function and tell it, okay, we want to run cyclic low S. And in parentheses, these are the available options. So we'll hit normalized data. There we go. And so the next step is to write the QC report. Again, we have options on the column grouping. So how do you want to color your PCA plots? Is there another factor you want to look at? Say you included a gender effect. Well, you can actually color your samples based on the gender and start to see, do I have another batching effect that needs to be removed from this report? So we put the QC report in the same folder, and now we have our violin that shows the overall normalized distribution by each sample. And then this is where you could use to see, okay, if you have a ton of data down here that are missing or 0, you may want to increase that filtering option that we played with earlier. The PCA will show you any samples that might potentially look like an outlier. So you may want to check on these 2 samples that are separating out in your PCA plot and from your cluster dendrogram. So in this case, you might go back up to the filter samples function and tell it to remove these 2 samples. So you have that option also within the report. Another one we include is Pearson correlation of each of the samples. This again will kind of show you how highly correlated your sample replicates are or not. And then lastly is a missing value table. And this is helpful to see, again, kind of that filter of proteins by missing data. Do you need to filter out more, or is it okay? This particular one, there's not that many missing values, and this is only plotting the proteins that have a missing value. This is not plotting every 8,000 proteins in your data set. So pretty good on our quality control. So now the next step, and this is really the power of this particular package, because if you're like me, kind of struggle a little bit with really having to think through the model matrix design for how do you want to set up your statistical design. So we try to make this process a little bit simpler to be able to run your analysis. And I'm going to make another video that goes into a lot more detail about normalization methods and a separate one about the Lima model designs. So for now, this is the way you would run it is we would use the add design function that's in this package. We tell it we want to use our normalized data that we've just normalized. And the design formula is a function that we've included where you literally just write the formula for the matrix for that design that you want to make. So the tilde 0 is the no intercept model, which is why I've called it no intercept, and plus group. Group, this is again, it has to be specifically typed as whatever you have in your sample metadata information. So this column is what I'm using to group my samples on to do my analysis. So we're going to run that. Now I have a new object called no intercept that includes the design formula. This is just building on this original object. I'm just kind of rewriting it so I don't get rid of my data if I need to go back to it. We still have the annotation, the metadata, and now we've added in our design formula. And so if we go back to here, if you want to see what that design looks like, you could say no intercept and then design. And then you can Control Enter, and that will show you how your samples that are included in this analysis, how they are flagged as either cancer group or normal group. And so these headings here are really important for this next step on this add contrast function. So now we need to tell the model how to compare these two groups. If you do a reference model without this tilde 0, it's just going to do it to the control reference automatically, and you don't have as much control of telling it the order of your fold change you want to calculate. So by doing it with a no intercept model, we can actually tell the algorithm how to calculate our fold change. And so in this contrast vector, we've got cancer versus normal. This first part is going to become the display on your final results and your plots, and we'll tell you what the comparison is. So this doesn't matter as much of if it's uppercase, lowercase, how you type it. But this part and the formula is important. This has to match those headers that are in your design, and that originally came from your sample metadata column. So however you have these listed in that meta information sheet at the very beginning really sets you up. So here I have cancer minus normal, and that's how we're going to now add our contrast. And now everything is included, so we can actually run the model, fit Lima model with our data type. And now we're going to extract all those results. And this is another important factor. Here we have our p-value threshold set to 0.05, and we have the log 2 fold change set to 1. So this will be a two-fold change criteria for all of your resulting plots. If you want to use 1.5 fold change, you would change that here. Or if you want to have a less stringent p-value threshold, these are the options where you would change that, and then it will change the coloring and what it considers significant in your final results. And that is here with our right limit tables. Run this. So we're now writing all of the tables. We're going to write them into a new folder. So instead of QC report, we now have a DA results folder. And so it's going to write individual contrasts. In this case, we only have one. So to have the CSV file in case you need it for downstream applications. But it's also going to create an Excel file. So this results Excel file will include all of the data. I'll have all the annotation data. Let's just go and see if I can pull this up. Okay. I've Got to go open this real fast. Sorry, I should have had this pulled up, but I did not. I'm almost there. Workshop. Okay. So here is my DA results. And then here's the summary will actually show you an overall significance list of your different comparisons. So in this case, I only have the one comparison that we're looking at. So it's only going to have the cancer versus normal. But it includes how many proteins were downregulated that passed significant p-value or past the adjusted p-value. In this case, none of them passed multiple test adjustment using these threshold values. Non-significant, this is everything else, and then these are the ones that were upregulated. But the results, going to open here. This is real life demo going on. So the results, we have our protein annotation. We have that UniProt ID column that's associated. We have all the protein information. So any protein annotation information we had is included here. The next set of data is your normalized intensities for each individual sample. And we do include in the header which normalization method was used, so you won't forget what you did when you ran ProteoNorm. Very, very helpful. And followed after that is the statistical output for this particular comparison. So we have our log2 fold change. It comes out of the Lima top table results. We've also highlighted any that were downregulated or upregulated. So down is green and up is red. Those are just basic Excel colors. And then we have the average intensity, so you can start to get a feel for the abundance of the protein. We have our unadjusted p-value and the adjusted p-value. And extra columns that we've added, this is a flag that shows if that particular protein passes both criteria. So in this column, you're looking at the unadjusted p-value. So yes, it's significant by p-value, but it's also significant by fold change, and it's downregulated. So that's going to flag it as a -1. If this was positive and upregulated, then we would flag this as a +1. So that way, when you sort through your table, you can get to that really top list with a higher stringency criteria, say adjusted p-value plus fold change, and you could sort and get to that very top list of your proteins. So the order of the label also corresponds with the fold change. So this showing a green down-regulated fold change means that protein is down in the first group listed, which is cancer. So those are actually down-regulated compared to the normal. So the order of this is really important, and that's how it works over here when we set up our contrast. So the order of your label here matches the order of how we did our comparison so that we understand how we actually calculated our fold change. So the table is really good. It's great to be able to do more plots, more downstream analyses, pathways, all of that sort of thing. But with this right lemma plot, which is our last function of this package, is going to write the interactive HTML files. And those are really super useful for just downstream looking at your data. So the grouping column, again, it's very important. This is how we're telling it what's cancer, what's normal. But the table columns, this is going to be where that at the very beginning, the annotation data that we imported into our data frame. So UniProt ID, I'm pulling this column, and I'm calling protein info. So this one. And what this is going to do is going to put these two pieces of information into our interactive plot. And then the title of the column, I'll show you where this shows up in a second. And then we're putting this in our DA results folder again. And then you can set kind of the height and width of your tables if you like. So now let's go back to our folder here. And now within this DA results folder, we have a new HTML report. So we'll open this up, and Google Chrome works pretty well. And this will show you the volcano plot, and it's going to show it to you using the adjusted p-values to start with. But you can change this and look at unadjusted p-values, and then the plot will change to that. We also have an MD plot for the mean difference plot. So there's 4 different options on how you want to visualize your data. But the really powerful thing is if you hover over each dot, each protein in the display, it'll show you that information that we told it we wanted. The UniProt ID, protein info, and then the stat output. But if you click on it, you will also generate a second plot, and this will show you each of the groups included in the analysis, and then every dot here is the different sample. So this is the normalized abundances for each sample that's included in this group. And then you can start to see the variability that's happening within the samples of your groups for that particular protein of interest. So the lower your p-value is, probably the more variable you have in your data. The more significant your p-value is, usually the better you're doing on your replicates in an agreement. And when you do change these, this scale does change a bit. So this is how you can start to kind of play around and actually just interact with your data. Again, if there's a protein of interest that you really want, you can just type it in here and click on, I just randomly picking one, and it will highlight where it is in the volcano plot and show you the results there. So it's an easier way to look at what's in that Excel file, but in a graphical way. And so again, when we did our code and we ran this lemma plots table columns, so UniProt protein info, that information is what's generating in this table for your HTML report. So with that, that is the majority of how to run an analysis. So if you wanted to go back and rerun analysis, change the model, add some other groups, change your comparisons, you can easily load it into this ProteoDA package and rerun your analysis. So we really wanted to try to focus on where the input is more of a downstream of any database search program that you use, because each, like MaxQuant, Spectronaut, they all have different formats that they're going to generate data from a database search. And so really from here, it's just you take that information, however you generated it, put it into the format of just a basic data frame with your sample abundances for all the protein abundances, and then you should be able to run each of those functions to complete the analysis. So with that, I thank you for listening to me talk about this package, and I wish you the best of luck with running your protein analyses. Bye.